Today in pulmonary physiology, we'll discuss some general integrated techniques to think about the pulmonary system and physiology as a whole. We will cover some key factors related to the mechanics of breathing. Then we will discuss airway closure and compression of the airway on expiration. And we will review some basic biophysical principles related to resistance and airflow patterns. Let's start first with our integrated approach or our model for the pulmonary system. If you break up the pulmonary system into some key subcomponents, you could think about it as bringing the air into the lungs from the atmosphere, bringing in oxygen from the atmosphere and expiring CO2, the process of breathing, better known as pulmonary ventilation, as our first point. Second, after the air makes it down to the alveoli of the lungs, it needs to diffuse across. The oxygen needs to make it onto the red blood cells, and the carbon dioxide from the red blood cells needs to diffuse back to the lung. And this diffusion is critical in adequately oxygenating your blood and maintaining the pH of your blood. Then once you have the right gas concentrations in the blood, number 3 is you got to transport that blood. So this is where your heart, both your right and left heart, come into play. And you'll need competent blood vessels to deliver arterialized blood down to peripheral tissues like muscles, the kidney, the brain, where the oxygen from the red blood cells then diffuses across into the tissue, into the mitochondria to make ATP through oxidative phosphorylation. And carbon dioxide stemming from the Krebs cycle gets released back onto the red blood cell. So there's like a reverse diffusion that relative to that which occurs at the lung. And then we deliver back through our venous system that blood, and then we do the whole process over and over and over again. So the way I like to think about this is to break up our discussions in each sort of subcomponent. So we'll start particularly today with ventilation, with breathing. And whenever you think about a system and the way it functions, it's always good to kind of take a bidirectional approach from understanding the way cells work and the way organelles within cells work, and trying to broaden the perspective to understand the whole person, the whole organism. And likewise, it's important to also work in reverse, where we try to think about clinical scenarios of the person and in a reductionist kind of way, go back and think, well, what might be going on with the cells or the organelles within given tissues and systems? The real trick in physiology and in pathophysiology is to try to understand systems integration against one another. I mean, certainly the GI system doesn't work in isolation. It's very reliant on the brain. And both of those systems are very reliant on the lungs and the kidneys and the heart and the blood vessels. So integrated approach, everything is everything. And that's important to understand. But as we delve deeper into each system, we can gain a lot of knowledge and some good methods of understanding physiologic situations by going back and forth in a reductionist to a whole human and back again in our thinking. So with relevance to the lungs, breathing, as I said, is called ventilation. Some texts refer to breathing as external respiration. Internal respiration means what's happening at the level of the mitochondria, and aerobic respiration. I prefer the term ventilation, and ventilation is often abbreviated as V sub E, the little dot on top, which means per unit time. Ventilation is equal to how much air one takes in with one breath, that's called the tidal volume, multiplied by the breathing frequency, how many times you breathe per minute. You'll see, and you probably already are well aware, that our one breath volume is somewhere around 500 milliliters, and we breathe somewhere around 12 times in a minute, and that gets us our minute ventilation level at rest. Both of these would go up, for example, in situations like stress and exercise. And the idea of what regulates our breathing, well, it's very complicated, and it's done so at the level of the brain. And we'll discuss that in and of itself. Let me just say that there are stimuli that are gas/chemical stimuli that primarily regulate our breathing. And if you were to rank order the stimuli of the factors that make us want to breathe more in sort of automated fashion, you don't have to think about, oh, let me breathe more. It's done for you. Kind of speed up your breathing based upon these chemical signals. The first one is carbon dioxide concentrations. When carbon dioxide goes up, increases in the serum, that stimulates the breath. The second is oxygen concentrations. If that goes down, that stimulates the breath. There are other stimuli, anything from muscle receptors and stretch receptors in the lung, etc. But these are the key two chemical signals that get sensed at the level of the brain, principally in the medulla and around and about the pons. And then based on those chemical signals, stimulates the breath. I should say oxygen is mostly sensed at the level of the systemic circulation by peripheral chemoreceptors. The ones up around and about the medulla are key in sensing CO2 and are very regulatory in trying to keep a stable pH in our bodies. So we breathe to maintain CO2 to maintain pH because we only have a narrow range. Now, while the process of breathing is neurologically automated, the lung itself is moved in a very mechanical sort of fashion. And basically, to make a long story very, very short, we bring air into our lungs by causing skeletal muscles like the diaphragm to contract. And this alters pressure gradients within the thoracic cavity and the lung and draws air in for inspiration and out for expiration. So when air comes into your body, it passes through the upper airway. And the components are listed here, the nose down to the larynx. It gets warmed, it gets filtered, and then it gets directed down into the lower airway, which starts with the trachea, which is just a big old cartilaginous tube that is a superhighway for airflow. And then that delivers the air down to the alveoli and the respiratory bronchioles, and it's these areas that allow for diffusion to occur. And the diffusion, as illustrated on a couple slides ago, states that this is where we have the transfer of gases from the lung into the blood and vice versa. So the upper airway has a very specific anatomy, and we will cover the regulation of that on a separate occasion. What I do here is I'll just pick up with the flow of air into the trachea. And the trachea is about 10 to 20 centimeters long in adults. Obviously, taller people have longer tracheas. It's comprised of sort of C-shaped cartilaginous rings 16 to 20. The back, I should have drawn it this way, the back of the cartilaginous rings is sort of missing because the esophagus runs back there, and you don't have the cartilage leaning on that. So the cartilage is hyaline cartilage. It's very protective against your quote-unquote windpipe. As you descend down the trachea into the branches of the right and left primary bronchi, the point at which you see this bifurcation is called the carina. And then you're progressively losing the cartilage balance of the airway. I mean, the airway has basically 3 layers as a whole. There's an external protective layer. In this case, in the trachea, for example, you've got the cartilage. It doesn't have appreciable smooth muscle, which is generally an area of the respiratory pathway. And on the inside, there's like a lining of epithelial cells that's sort of the Teflon that allows for a smooth airflow. So maybe the trachea is not the best example for this, but in general, the 3 layers are the external layer, the smooth muscle layer, and then the internal epithelial layer that has relatively different distributions throughout the respiratory tree, the ventilatory tree. When you get down to respiratory bronchioles and whatnot, they're very much smooth muscle and epithelial cells, and obviously not any kind of cartilage. So generally, the way it's set up is that where you have high flow rates and high potential for turbulent flow, you've got like sort of backup anatomically in the system. And as the flow becomes more layered or laminar, you need less of that outer layer to keep everything sort of together, and you get more into thinner layers of smooth muscle. And then, the alveoli are just basically epithelial cells with a very thin membrane that favors diffusion. So if you take the trachea as being like a tree trunk, like in a reverse tree, that's like generation 0. When you break off into the primary bronchi, that's the first generation, then you break off in the secondary bronchi, so on and so forth. About the first 16 generations is called the conducting zone of the lung. This table gives you some of the anatomical features of those areas of the lung. This is just a place where air moves back and forth, conducting. But when you get to about the 17th generation, appreciating that the alveoli are the 23rd generation, there's some gas exchange. Hence, the respiratory zone is what it's referred to between the ventilatory tree and the blood, and you can appreciate some of its sizes. Now, you literally have millions upon millions of alveoli, so there's a tremendous surface area, and there's a lot of blood vessels in the lungs, so you have a wonderful total area for diffusion. If you look at the model in terms of the airway generation and stages of branching, the resistance to airflow is highest kind of on the top of the system, somewhere around the 5th, maybe 6th generation or so, and then it progressively falls as we get into the alveoli. And the reason for that is that you just have such an incredible number of these respiratory zones that whenever you put resistors into a parallel arrangement, which is the case here, that causes a very drastic reduction in resistance. And we'll talk about resistance in a little bit. Anyway, this is the way the system's anatomically distinguished. And when the air gets down then into the bronchial tree, one of the things I'd like to just comment on is the right and the left main stems of the bronchi are at slightly different angles. The left main stem's at a 45-degree angle, whereas the right stem's at about a 25, 30-degree angle. 3 branches on the right, 2 branches on the left. And then that leads to the progressive branching again down to the alveoli for gas exchange. So the conducting function of the bronchial trees in these upper areas are as prominent in distributing the air across the lung the best that it can. And then as you get deeper into the bronchi, as I said, there's more smooth muscle, and then whether or not that smooth muscle contracts or not will determine where the air goes, what alveoli will see fresh air. Here's a micrograph of alveoli, and you can see they kind of look like little grape clusters. All the little A's stand for alveoli. The D is an alveolar duct leading into these alveoli, and you can't really see pores here so easily, but there's connections between adjacent alveoli. So you have these little grape-like clusters that fill with air and can share air among their neighbors. So they work as kind of a collective unit. The pores allow some of that connection. And literally hundreds of millions of alveoli in those lungs. And then you can have problems with that. You can lose alveolar number with pulmonary disease, infection, pulmonary embolism, those sorts of things. And that becomes a problem because then your gas exchange becomes problematic. Notice the walls of the alveoli are very, very thin, favoring diffusion. Here's a pore of cone. And you can almost just draw in your own little capillaries around these alveoli. They have many, many capillaries, again, well suited for diffusion. When we breathe mechanically, inspiration is about 1/3 of the cycle. And inspiration occurs when the diaphragm gets pushed down into the belly, and that draws the air into the lungs. The diaphragm is a skeletal muscle. It's a slow twitch red muscle. It is innervated by the phrenic nerves, and as I said, chemical signals around and about in the brainstem trigger the phrenic nerve to make action potentials to make the diaphragm contract like a typical skeletal muscle does. Now, there are some other muscles that help with inspiration, like your external intercostals, your sternocleidomastoid can. It's an active process, so it requires muscle contraction, it requires energy consumption. Expiration, on the other hand, takes about two-thirds of the respiratory cycle, the ventilatory cycle. It's passive. Expiration is at rest, not reliant on skeletal muscle. If this is an alveolus, there are 2 factors that make the air get pushed out of the alveoli, out into the atmosphere. And those 2 factors are the elastic nature of the lung. Like a rubber band, when you stretch the lung out, it snaps back, and that helps push air out. And then the second factor, and we'll cover these throughout our discussion today, is the surface tension. Surface tension in the lung is a tension at the water-air interface. There's some water vapors inside the alveoli, and they're attracted to other water vapor molecules, and that causes the lung to want to collapse. So you can kind of picture the alveolus snapping back on itself and pushing the air out because it's changing the pressure when it does so. At rest, this ratio of inspiration to expiration is a 1-to-2 ratio, and you can change those ratios with mechanical ventilation, which is very relevant to you all. You can change it with exercise, disease, etc. So there's a lot of plasticity in the way we breathe, and for now, let's just appreciate expiration at rest is passive. Inspiration at rest is active, but when we start to undergo increased breathing, like with exercise and with disease, then expiration might actually become active, and our belly muscles, like our rectus abdominis, becomes involved in trying to push the belly and cause an upward dispersion of the diaphragm to change the thoracic pressures, etc. This is a nice illustration of the diaphragm ascending into the belly here, and that's what draws the breath in. It's a thin, dome-shaped muscle. It's attached to the lower ribs. As I said, the phrenic nerves on both the left and right side, which have their roots at the cervical segments 3, 4, and 5, control the diaphragmatic contraction. It's typical skeletal muscle action potentials, releasing acetylcholine, causing the muscle fibers to contract. You can have injury to phrenic nerves. You can have injury to the spinal cord. This could impact half or all of the phrenic nerve innervation to the diaphragm, and then you would be really with a problem because you'd have a difficult time breathing. Again, other muscles can help you breathe, like the external intercostals for inspiration, sternocleidomastoid, but the diaphragm is the prime mover here. And that's point number one. Now, if you look at the cartoon from the side, the other thing you might want to try to envision is that the ribs get lifted up and out. The whole thorax expands. And the purpose of the thorax expanding and the diaphragm protruding down into the belly is it makes your thorax just bigger. And when it makes it bigger, that lowers the pressure in some of the components of the thoracic cavity, and that's what actually draws the air into the lungs. So in a normal breath, as you're just resting, your diaphragm just moves a little bit, like a centimeter. This is not a minus sign, this is just a dash. About a centimeter it goes down. And then if you're breathing more vigorously, like in stress or exercise, you can make it contract more vigorously and it'll fall 10 centimeters. Now, because the diaphragm separates the abdomen from the thoracic cavity, if you were to paralyze the diaphragm, and we're going to talk about the negative pressures, like a vacuum, that occur in inspiration in the thoracic cavity, so I'll put a minus sign here. When the diaphragm is paralyzed and you draw an inspiration, the diaphragm will actually get sucked up into the chest because of the vacuuming effect, pulling it upwards. It doesn't have the muscle tone that it otherwise would have. And so that's very interesting as well. And I can't emphasize that being a slow twitch muscle, the diaphragm can't get tired. I mean, it can get tired, but it really can't rest. We're always breathing through our entire lives. People that have to breathe more vigorously with pulmonary disease, chronic obstructive disease, etc., or even more so in restrictive diseases, this can cause the ventilatory muscles to fatigue a little bit. So that's where we would need things like conditioning, exercise conditioning, to try to help their endurance and their strength and their force development, etc. So, as I said, other muscles of inspiration, which are listed here in these figures, are the external intercostals, and they're attached to the ribs. They slope downward and forward, and what they do is they pull the ribs upward and forward, again expanding the thorax, allowing this sort of bucket handle approach, which you'll see in one of the subsequent slides, to provide movement of the ribs and make a negative pressure ultimately so that air can flow into the lungs. The intercostal muscles are supplied by their own intercostal nerves that stem off of, these are alpha motor neurons that come off the same levels of the spinal cord that's appropriate for the level of the skeletal muscle. And if you had nerve injury or if you had intercostal muscle injury, in this case, the external intercostals alone, it would not severely impact your inspiration because, again, it's not the prime mover, the diaphragm is. This is the bucket head, the bucket handle approach. If you can kind of picture the bucket handle moving back and forth, that's kind of how your ribs go with inspiration and expiration as an axis of rotation on your vertebrae. These are the muscles of, I should put, forced expiration. Remember, passive expiration, the elastic quality of the lung and the surface tension is what does that. But in forced expiration, we push our belly inward, if you will, with muscle contraction of the transverse abdominis, the internal obliques, the external obliques, the rectus abdominis, and also the internal intercostals. So when we're hyperventilating and ventilation expiration in this case becomes active, these get active and a strong core, if you will, or abdominal muscles push in and change the thoracic cavity pressures, making them more positive, and that forces the air out into the atmosphere. Let's not forget about green plants here, right? Our oxygen comes from green plants, and then we inspire that oxygen and we, use it in our aerobic respiratory pathways. And then our byproduct of CO2, as we expire, it also goes to green plants to spark on photosynthesis for the production of oxygen, essentially. And so there's a symbiosis between green plants and human beings, and our atmospheric oxygen and our atmospheric CO2 need to be well balanced so that we can kind of stay alive here. Nonetheless, expiration and forced expiration, work is involved, and you can also activate these belly muscles in coughing, in vomiting, and in defecation, in Valsalva maneuvers, those sorts of things. So if you take your thoracic cavity, the blue being your diaphragm, and you look at the thoracic cavity, appreciate you've got your great vessels in your heart kind of in the middle, right above the diaphragm. Right side, you got your liver, in the belly. And then on the rib side, you've got these two layers of tissue, the pleural tissue, the parietal pleura, like on the underside of the rib, and then the visceral pleura is what is like the tissue right on top of the lung. There's a little space there called the pleural space. It's a potential space, but it has some fluid circulating through it. That's the pleural fluid. And it's this space, this area between the rib cage, if you will, and the lung itself. This is the area that's so important in drawing the breath. Now, if you take a better illustration of that pleural space, this illustration is a nice one. If you blow up the pleural space right here, you can see here's the parietal and the parietal and the visceral pleura, and you can see this space a little bit better. And the pleural fluid that floats through there is or needs to be pretty well regulated in terms of its volume. It's somewhere about 0.25, 0.26 mils per kilo of body weight. Circulates around and about the lungs. It lubricates the lungs, lessens the friction. It's protective on the lungs, and it's an added barrier on or for this very important system. And the pleural fluid turns over quite frequently, and it drains into the lymph. And certainly when you're producing pleural fluid, you need to drain it out at equal volume. Otherwise, that pleural fluid will start to increase. Now, that would be a problem, as you might see in something like a pleural effusion. Lung infections, left-sided heart failure, sometimes trauma, these sorts of things can increase the volume of pleural fluid. And then that volume starts to build up, and that can actually start to compress on the lung and make it that much more difficult to breathe. And you can partition the intrapleural space where that fluid is circulating from the lung itself, the alveoli, the inside of the alveoli. And side by side, this illustration does that to some extent, where At the end of expiration, you're looking at a cartoon over here. Let's call it A. And then at the inspiratory maneuver here in B, and I want to just bring up a couple of key areas, if I may. So the first thing is the intrapleural pressure, the space between the parietal and the visceral pleura, has a pressure in it based upon that pleural fluid, and based upon the fact that the rib cage wants to bow out, it wants to move in this direction, and the lung, because of the elastic tissue and the surface tension, wants to bow in. So like the two areas like get peeled apart, and then there's fluid in there. And that makes a negative pressure relative to atmosphere. What I'm trying to say here is if you call atmospheric pressure 0 centimeters of water, relative to the atmospheric pressure, there's a vacuum in the intrapleural space. Its pressure is -5 centimeters of water. Now, every millimeter of mercury is 1.36 centimeters of water. So you can convert millimeters of mercury and centimeters of water just to give you appreciation for that. So there's a negative pressure there, and that's what keeps the lung open, if you will. When you're not bringing in any air from the atmosphere into the lung, the pressures inside the alveoli and the atmosphere are the same. They're in equilibrium, 0 centimeters of water, 0 centimeters of water. When your diaphragm contracts and moves down, what that does is it makes more room, if you will, in the intrapleural space. And it makes the intrapleural pressure become more negative. So in this example, it fell from -5 centimeters of water to -8 centimeters of water. See, the space is bigger. And that made a bigger negative a vacuum, if you will, and the lung got, or was provided a force differential where it was able to get pulled out into that vacuum. And when that occurred, the pressure inside the alveoli actually falls. In this case, it fell to -1 centimeter of water. Well, the atmosphere didn't change. That was 0. There's -1. Air moves from a higher pressure to a lower pressure. So we went from 0 to the lower pressure, and now we fill up the lung with the air. So the thing that made the alveoli take the air in, the mechanism that allowed the alveolar pressure to fall, is the drop in the intrapleural pressure generated by the skeletal muscles, like the diaphragm, that allow for an expansion of the thorax to change those pressures. Now, clinically, you know that could get altered with issue of pneumothorax. So here is a similar drawing showing you the interrelationship between the pleural pressure and the alveolar pressure. But if the pleural pressure is exposed to atmospheric pressure with some sort of a trauma or a hole or something, air gets into that space. Now it's no longer a vacuum. It causes a positive pressure and causes the lung to start to collapse. Then it becomes very difficult to draw the breath in. So you all are well aware that you can then put in some sort of a vector to drain the air, like a chest tube, get the air back out, and not try, with the attempt to try to expand or re-expand the lung. This is often the case with surgeries, thoracotomies, and whatnot. Your patients have chest tubes to try to, when applied to, a suction device, to try to make a negative pressure to try to pull those lungs open. The other place where you often use chest tubes is to drain excess pleural fluid, which might also make for positive intrapleural pressure. So by looking at pneumothorax, I think we can all gain a great appreciation for the significance of what the negative intrapleural pressure does to the lung. And if you prefer millimeters of mercury across the breathing cycle, maybe an approach like this would work. Here's at rest where our atmospheric pressure is 760 millimeters of mercury at sea level. Our intrapulmonary pressure, think of that as the alveolar pressure in here. That's, in this example, also 760. There's no differential, there's no delta, 760 to 760, so there's no airflow. So we're quiet. The intrapleural pressure is 756, so that's negative. And we have this, and I don't love this term, the difference between the intrapulmonary and the intrapleural. It's really a transmural pressure. The difference between the two is 4 millimeters of mercury. Just kind of avoid that at the moment. You can think of it instead as say, okay, there's 4 millimeters of mercury of suction keeping my lung open, but I don't have airflow. Now we move and the diaphragm contracts. That lowers now the intrapleural pressure to 754 from the 756, so it fell by 2 units. And the alveolar pressure falls from 760 to 757. So now we have 760 outside, 757 inside. There's a delta of 3. Air flows in. The change in pressure, the delta pressure, is the driving force to bring the air into the lung. Now, of course, as air fills up the lung, that pressure difference will be, mitigated, but that's the driving force during inspiration. And then during expiration, the diaphragm returns back to its normal configuration, its resting configuration. But again, the alveolus, because of its elastic qualities and because of the surface tension, causes the alveoli to have an inward pull. And then that causes the alveolar pressures to be 763. This would be in quiet expiration, 760. Now we have a delta, but there's more pressure inside pushing the air out. And the intrapleural pressure stays at a good old vacuum of 756. So if you put all of this together and think about the understanding of the various pressures driving the breathing cycle, you come to this very important figure from the John West book. And it's a complicated graphical system where if we look at the x-axis, time is on the x-axis. And then there's panel A, panel B, panel C, and panel D. And each one of these represent a different dynamic in the lung. The top panel represents the tidal volume. So it's in liters per minute. Now what this is showing you is that ventilation, as I said, is the tidal volume times the breathing frequency. When you draw your inspiration, air flows into the lung. So this is flowing in at 400, 500 milliliters during the inspiration. And then when you expire, and you can measure this in a spirometer, you blow off 400 or 500 milliliters. That would be your tidal volume. Okay, and that's graphed here. This person's breathing into a spirometer, and you're actually getting a graphic. They present it in an inverse way because the air is getting blown in from a container into the lung, so it goes down, and then you blow it back into the container, and it goes up, and that's the nature of this curve. So that's A, in liters per minute. And if you look at the way the air gets into the lung in terms of its flow rate, which is expressed in C, you see that the flow rate is sort of the speed, the volume per time. It peaks. In other words, the greatest volume during the inspiration per unit time is in the middle of the maneuver. And that's also the case for expiration. In other words, the air's not all coming in at the same rate. It comes in a little bit, it peaks, and then it slows down towards the end of inspiration. At expiration, it's slow, speeds up, and then it slows down again. And the thing that controls the flow rate of air coming into the lung is the alveolar pressure. So the alveolar pressure is in equilibrium at the beginning of inspiration to the atmosphere. But then it gets more negative because the intrapleural pressure falls. And in doing so, it reaches its most negative pressure about midway through the inspiration, and that's what draws the added flow rate. And then that slows down. Like it looks like it. Look at D and C. They're mirror images of one another. The alveolar pressure is a mirror image of the flow rate. We skipped B, but let's go to it because that's the intrapleural pressure. As illustrated on the previous slides, and you can just look at the solid line here. What the solid line shows you is the intrapleural pressure when it's also considering or accounting for the alveolar pressure. During inspiration, our intrapleural pressure goes down and down and down and down, -5 to -8. Well, it's that -5 to -8 that makes the alveolar pressure go down, that draws the breath in to bring the volume in. And then on expiration, the intrapleural pressure gets higher and higher and higher because the diaphragm and the external intercostals are, returning to their normal configuration. Goes back to -5. Still negative, still a vacuum. But that then causes a differential to push the air out in combination with the elastic forces and the surface tension for expiration. The point of the story is the muscles control the intrapleural pressure for both inspiration and forced expiration. That, in turn, from B, controls the alveolar pressures through a transmural pressure differential. That causes delta pressures from the atmosphere into the lung, dictates the flow rate, and then air enters or exits the lung in inspiration and expiration, respectively, and that dictates the volume, that dictates the tidal volume. Our brain, stem, and chemical stimuli regulate this for us, principally by the phrenic nerve activity and the intercostal nerve activity. When you're high in CO2 and low in O2, it says, hey, got to breathe more. So this will happen more frequently and with greater pressure differentials. It's really an important understanding to the mechanics by which we breathe. Now, I would ask you, just as a critical thinking exercise, to just, you could pause here for a moment and say, well, what would happen if I mechanically ventilated this patient, if I forced from a machine air into this airway with a positive pressure? What would some of our considerations be in terms of, say, flow rate, alveolar pressure? Do we then need an intrapleural pressure to be negative in order to sort of shove that air down into the lungs? Wouldn't it be great if you just had a machine that pushed a whole bunch of PSIs, into the lung? Or are you worried about, the lung? Think of it almost as like a balloon, like that's an alveolus/balloon. Can you blow it up too much and actually cause barotraumas and whatnot? So we've got lots of ways that we can mechanically induce breathing with incredible technology that can regulate so many aspects of the way the air gets in and out. But how does that differ from a typical innate breathing situation? That would be nice to sit back and mull over. When a person is breathing on their own, it costs muscles to do work. And the muscles are doing work based upon how much pressure they're generating versus how much volume gets moved in and out of the lung. So you're moving more air in, say, exercise, like this person just exercised, and they're making more negative intrapleural pressure, so there's greater excursion of the diaphragm and whatnot to push the air into the lung and whatnot. And there are really well pulmonary physiology pressure volume curves that can help us look at the work of breathing. I mean, this is the formula, but it's broken down into components. And if you break the pressure and the volume into the components of the biophysics that the lung has to overcome, it has two subfactors involved. The first is the elastic force of the lung. You have to overcome the elastic force of the lung to draw the air in. Like a rubber band, you have to be able to pull it open. And the second force is the viscous force. The lung like rubs against itself as you're breathing, and as it's leaning on itself, shearing against itself, there's also work involved with that. Far and away, the elastic work is the most work that the lung does. That's illustrated over here in a pressure volume curve. Now, a pressure volume curve, on the x-axis you have the intrapleural pressure. On the y-axis, let's just simplify this and call it volume into the lung. And you breathe in, air comes into the lung, A, B, C, as the intrapleural pressure falls. We fill up the lung, the diaphragm returns back to its normal place. We start to expire CFA, and at every given level of intrapleural pressure, there's a little more air left in the lung during expiration than there is at inspiration. And this is drawing a tidal volume and whatnot. Now the area, and I'm going to color it in here, everything under the area under the curve that I'm coloring in to the solid line right here, all the way over here, during inspiration, represents the elastic work to bring air in to pull the rubber band open, to pull the lung open. Most of the work is done to do that. Of the viscous work, which is the pink lines already on the figure, that's far less work. So we need to pull the lung open. That's most of our work. And so when you're trying to draw a really deep breath, as you pull the rubber band, doesn't it get harder as it gets more and more stretched? Same thing with your lung. And so that helps regulate how much volume gets moved and how much pressure we need to generate, in this case a negative pressure, to move that volume. So that's the work of breathing, elastic forces and viscous forces, with elastic being the biggest. The higher the breathing rate, when people are breathing very fast, the higher the viscous work. And as I just alluded to, the higher the tidal volume, the higher the elastic work. So there's a balance, like everything. There's a sweet spot. The 500 mils, the 12 to 15 breaths per minute is our sweet spot. But then you've got patients that have changes in the phenotype of their lungs. Their lungs might get stiff or have a reduced compliance. And what they do is they start to breathe in smaller, more rapid kinds of breaths. then there are other kinds of patients that often have severe airway obstruction, like a chronic bronchitis or something. They tend to breathe more slowly. And their breathing patterns are attempts to normalize the breathing work and allow it to be a more efficient, a less costly endeavor. And that's something you can tell just by the way that people breathe, just by sort of watching them. The work of breathing, there's a whole literature on this, is the work of moving the lung and the chest wall. And studies in ventilated patients and paralyzed patients have been done. And the way it's calculated is how much ATP does it cost to breathe? And you can indirectly guesstimate ATP costs by how much oxygen a person consumes, the VO2. So you calculate the useful work that's done as the efficiency based upon a person's oxygen consumption needed to breathe. And then you calculate the efficiency, and you can get some idea that the efficiency isn't great. Of breathing, it's like 5 to 10%. But we don't use a whole bunch of our total body oxygen consumption / ATP to breathe. Generally, it's pretty low overall energy utilization to breathe. But in someone that has a lung disease, or perhaps is physiologically in a hyperventilatory state, then the respiratory muscles, the ventilatory muscles, they can start to use much, much more energy to breathe, up to 30% of the total energy cost of the body. So disease lessens the efficiency, increases the cost, and this is where people start to get tired, and it gets tougher and tougher to breathe as they become more and more short of breath. Now, the way you can think about the anatomy of the lung in terms of like the rubber band, like, do you have a rubber band that's pliable and easy to pull apart? Or do you have one that's really, really like an office rubber band, those big thick ones that's really stiff and hard to pull apart? That same sort of analogy pertains to the pressure volume loop. So here's a pressure volume loop drawn a little differently than what you saw in the work, but it's the same principle. On the x-axis, you've got pressure around the lung, the intrapleural pressure. On the y-axis, you've got your volume going in and out of the lung. Inspiration is illustrated with this line. Expiration is illustrated with this line. The lines don't match up. That's called hysteresis. At every given level of intrapleural pressure, there's more air in the lung on expiration, maintaining a residual volume, than there is on inspiration. And you can play games with the pressure volume loop, which is called a compliance curve, looking at different areas of the curve and calculating slopes, rate of rise, etc., and then dictate some characteristics or establish some characteristics of the lung. And so we're going to talk a lot right now about compliance and pressure volume curves. As I said, the hysteresis is the difference between the two curves. We have the lung with more air in it at the end of expiration. That maintains a volume in the alveolus, and that air helps keep the alveolus open, which is ultimately a good thing. The reason for that is, as we'll learn, gas gets trapped into the airway. We get airway closure and airway trapping. But it turns out to be, I hate to use the word good or bad as qualitative, judgment, but it's a quote unquote good thing here because it helps keep the lung open, and that's a good thing. The reason that there's hysteresis was interestingly established in experiments way back when, This is published in like 1965, showing in isolated lung preparations. Here's an inspiration and expiration, breathing air. And this is a lung preparation, breathing saline, inspiration, expiration. When you have an air mixture coming into your lungs like we do physiologically, there is a surface tension. The surface tension being tension between the water molecules and the air, where water and water want to bind, and that causes the lung to want to collapse on itself. If you perfuse the lung with just saline, physiologic isotonic saline, you get rid of the surface tension because there's no water-air interface, it's just all liquid. Hysteresis then goes away. So the conclusion was, why is there hysteresis? Well, there's more air in that lung because of the airway closure. Yes, that's something. But then from a mechanistic point of view, the surface tension in the lung, when you wipe it out, it obliterates the hysteresis. And hence, that becomes something to greatly consider. Again, the air has surface tension. This one, not, less so. So the surface tension's got to be mechanistically involved here in establishing the hysteresis. So it's the air and the surface tension. Now, that's just like an overall view of the pressure volume curve, and back to the pressure volume curve. X is the intrapleural pressure, Y is the volume going into the lung. The compliance is, and we all need to know this, is the volume entering the lung per pressure change. So it's the Y per X. Now our lungs are, in normal instances, are very compliant. You can get 200 mils of volume per every centimeter of water change in intrapleural pressure. And when you measure compliance in a human, you can measure it on expiration or inspiration. You can pick one. And again, because there's hysteresis, they're not the same. They're not synonymous. If you look at the inspiratory curve here, it's kind of S-shaped. So I want to appreciate that. And we work in life in this range of the curve, about 5 to 10 centimeter, negative 5 to negative 10 centimeters of water. And we're very compliant. You see the slope of this curve right here? The slope of the curve is, for just a little bit of pressure change, we get a lot of volume coming into our lung. But if you looked up here on the inspiratory curve, look at the slope up here. For a pressure change, we don't get much volume. So we don't have the same compliance throughout the entire curve. It depends upon where you want to look. So that's something that I'll speak to. Moreover, there are diseases that change the compliance. You can have a reduced compliance. That means you get less volume in for every pressure differential. And these diseases are restrictive, like in pulmonary fibrosis or pneumoconiosis, miner's lung, black lung, in ARDS, in autoimmune diseases like lupus, rheumatoid arthritis, and others, TB, alveolar edema, low lung volumes, not having air in there where the lung is collapsing, very difficult to, very stiff lung to refill with a volume. Or even left-sided heart failure where blood backs up into the pulmonary vasculature and you get some edema around and about the alveoli. And that leans on the alveoli, making it less compliant. So think about it, like as a rubber band, do you want a nice, easily open, pliable rubber band? Or do you want a stiff, reduced compliant rubber band? You want the more easily compliant rubber band. Reduced compliance increases the work of breathing as per all of these diseases. And that makes it miserable for those patients. If you took the normal, so let's just say, we're just going to call this, I'm just going to change it. I'm going to call it the intrapleural pressure. It's the same principle on the x-axis. and this is volume on the y-axis. And you looked at the idea of the normal pressure volume. Here's the change in pressure for the change in volume, and you can take the slope of the line. Now this was likely measured on expiration. See, it's more of a rounded curve, less S-shaped. And you went to a patient that had fibrotic lung disease. They're working on this line. There's reduced compliance. It took more pressure to get the same amount of volume. That made breathing more difficult. Now, the flip side can occur. You can have lungs that have increased compliance. And the two classic examples of increased compliance are emphysema and an aging lung, where in both instances, what you have are these big old floppy alveoli. So that aging, here's a normal alveolus. This big floppy alveolus has lots of air in it, big volume, and it doesn't take much pressure to make that volume. So it's a big volume per pressure change. It's more compliant. It's still disease-like. I'm not arguing like that's a good thing. It's different and distinct from a reduced compliant lung. You'll hear me and you'll see literature where you hear the term stiffness. Stiffness is the inverse of compliance. A lung that's compliant has low stiffness. A lung that has a lack of compliance has great stiffness. What causes the lung to change its stiffness? I keep drawing this picture of an alveolus almost as our simple model of the entire lung. Again, think of like, what if you changed the connective tissue around it? What if there was a lot of collagen around it? What if there was a lot of fluid and junk around it? It'd be harder to draw it open. So the elastic recoil of the lung can change. In aging and emphysema, the reason the lung gets all dilated and floppy, if you will, I use the analogy of grapes. Like nice fresh grapes are like taut, well formed, and let the grapes sort of get old and start to rot, the grapes start to get dangly and they lose their integrity. Well, that's what happens with emphysema. The connective tissue starts to change around the lung and it starts to dangle, allowing a lot of volume to be stored in there. It's bad volume, it's old volume, it's stale volume, but it changes the compliance. So you change your elastic recoil and you change your ability to expire that air, because that's one of those factors that determine the passive expiration. Again, elastic changes in the lung and the surface tension. So I mentioned before that those elastic changes in the lung versus or relative to the elastic tendencies of the rib cage dictate the intrapleural pressure. The rib cage wants to go that way. The lung wants to go this way. And that's what makes that negative intrapleural pressure lost with the pneumothorax. Not only will you change your breathing compliance with lung-based diseases, but if you had some sort of a musculoskeletal issue, postural changes, kyphosis, musculoskeletal disease, where your rib cage was less apt to bow out, to expand, that would also change the intrapleural pressure, and that too can also make it more difficult to breathe. So you have to think of the rib cage as well, outside of the realm of pneumothorax. This is just a nice figure showing at the air-water interface. Water molecules want to bind. Like if you ever watch rain, little driplets of water want to fuse into one. There's an attraction, a molecular attraction. And if you take and humidify air that makes it deep into the lung, with warming, those water molecules want to pull together. That causes and they're adherent to the sides of the wall, and that's causing the alveolus to want to collapse. Again, like the elasticity. And this is an okay thing. It helps us with expiration. It is what it is. It's like a soap bubble likes to contract to get to just its sort of right configuration. Well, the same thing here in the alveoli of the lung. But you can have an extreme where there's too much surface tension, and in the extreme, the lung collapses, the alveoli collapses. Here's a collapsed one, here's a normal one. Too much surface tension will cause the collapse. Now it's hard to get air in on the next breath. We have in our lung these epithelial cells that are of type 2 nature that secrete surfactant. You know about surfactant. It's like a detergent, and water likes to bind to water, and surfactant gets in the way. Now there's lipid in the surfactant. The lipid is called dipalmitoyl phosphatidylcholine, DPPC, and that changes the attractive forces between the water molecules and actually cause sort of repellent nature here. It keeps the water separated, so it lessens the surface tension and keeps this open. Now, this is very, very important because we can have pathological situations where we don't have adequate surfactant and we get alveolar collapse. The way the surfactant is synthesized is that you have these little vesicles called laminated bodies inside these type 2 alveolar cells. Here's one. Here's a micrograph. And they rupture and they release the surfactant out into the lung out here to keep the water away from water. Appreciate this is magnified 10,000 times. And this surfactant that gets secreted from these laminated bodies relies greatly on fats, on lipids that get delivered through the bloodstream. Now, there are two situations that I'll comment on in this next slide that can cause significant reduction in surfactant and make the lung more prone to atelectasis/collapse. The first one is on the back of how it's synthesized from fatty acids. If this is your alveolus over here, and this is a blood vessel that had a pulmonary embolism, you can't deliver the fats to the type 2 alveolar cells, so now you can't make and manufacture the surfactant. So these alveoli distal from the embolism will be susceptible to high surface tensions and closure. So adequate blood flow is needed to keep areas of lung healthy to manufacture surfactant. The other one is, and you know this, those of you that come from a PEDS background, fetal life. In fetal life, surfactant is formed relatively late in fetal life. And if you get a preemie without surfactant, their lungs, their alveoli are apt to close. That can induce respiratory distress and even death. We've got, synthetic surfactant that we can squirt down into the lungs to try to keep those lungs open until the baby can manufacture their own. So, surfactant is incredibly important to keep the alveoli open, to maintain for good surface area for gas exchange. It's compatible with life. And so it keeps a low surface tension in the alveoli. The other thing that surfactant does, and it's often less appreciated, is as indicated in bullet number 3, it keeps the alveoli dry. Without surfactant, fluid, can migrate into the alveolus. And the surface tension will draw that water in. By mitigating the surface tension, surfactant keeps the lungs from getting wet, from drawing fluid from the capillaries and the extravascular space. So you get less transudation of fluid into the lungs. And that's obviously beneficial because oxygen doesn't like to swim through water in the process of diffusion. That's a limiting factor in terms of diffusive forces across the lung. Now, I'm off on a couple little tangents on either way, but I think you get the spirit of the nature of the lung and you appreciate the significance of compliance. To take the story a little further, compliance in different areas of an upright lung, upright meaning sitting up, standing up, anything that's not supine or prone or recumbent, that's an upright lung, can induce regional differences in ventilation. Right to the story. We ventilate, if this is a lung, more air goes to the bottom of the lung than it does on the top of the lung. That's bottom line. And the reason for that is because there's a differential in compliance, greater compliance on the bottom of the upright lung than on the top of the upright lung. So why is that the case? Well, I think that's illustrated nicely on this figure from the West book. Here's a lung. Now picture this lung in an upright position and the weight of the lung, gravitational pull of the lung, pulling it down on the bottom. It's collapsing down on the bottom. It's leaning into the intrapleural space, if you will, making the intrapleural pressure, in this example, -2 centimeters of water. At the top of the lung, because the lung's dangling down, it's got an intrapleural pressure that's more negative, -10 centimeters of water. So the bottom of the lung is working on this slope of the inspiratory compliance curve. This is inspiration. And the top of the lung, follow the arrow, is working here. That's the top. This is the bottom. Which one has a steeper slope? Certainly the bottom does. So because of the upright weight of the lung changing the intrapleural pressure, we are working on a greater compliance. More air can enter there. Greater regional ventilation. Compliance less on the top. Less air enters there. And then the middle's the middle. It gets like a middle version. And that's what's spelled out for you on the wording part of this slide. So ventilation in an upright lung is greater on the bottom. So let's reflect for a moment. What would happen if you were lying in a supine position? Do you suspect that ventilation would be different on the anterior portion of the lung versus the dorsal portion of the lung? How about in the prone position? Lie on your belly. What if I lay with my left lung down? Would there be a difference, do you think, between left lung air ventilation and right lung? Dependent versus independent lung? Vice versa in the right lung? How might compliance and gravity help explain those phenomena? And the same thinking that we just went through here would be critical. There are situations on the base of the lung where you have such a low volume inside the alveoli, and there could be higher intrapleural pressures. Like maybe you've got a little bit of an effusion or something down on the base of the lung. Well, now the intrapleural pressure is actually creeping above atmosphere. It's no longer a negative pressure. It's a positive pressure. Well, heck, if this is your intrapleural space and this is the lung in that area, and now you're pushing on the lung, it's going to push air out. And it's going to make it much more difficult to bring in air. You're working down here on this portion of the curve. That's a real problem. That might require mechanical ventilation if that was, a widespread phenomena across the lung to adequately ventilate the lung. Yeah, you got millions of alveoli, but the air going to individual alveoli is heterogeneous because there are regional differences in compliance based upon the effects of gravity in the lung. And that would be applied to upright and non-upright lungs. So there's some really important principles associated with breathing mechanics. The next phenomena I'd like to talk about is airway closure, very relevant to expiration and keeping air trapped in that alveolus, maintaining a residual volume. So particularly in the base of the lung, but it's true throughout, you don't get all the gas squeezed out. The reason you don't get all the gas squeezed out, if I draw my alveolus here and I'll draw a longer respiratory tree, is that the respiratory tree gets pinched off. Like you make a dam here and all the air, not all, but some of the air that was otherwise trying to get out gets trapped. And this generally happens in the respiratory bronchi. They close first, trapping gas, down in the distal generations. In elderly people or in situations of emphysema where you lose elastic recoil, this even happens deeper down into, closer to the alveoli, and more air becomes trapped. And this can become problematic. It might require things like a submaximal gas exchange and might require supplemental oxygen and or ventilation. So airway closure happens to all of us, but it's more significant in folks that have a disease and with age. And no matter how hard you try, you can't blow more air out of your lungs, as is illustrated in this maneuver. What this person did here is they filled their lungs up with air, and then they breathed out. So air went. This is flow rate, out, 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 out. It peaked over time or over a volume of expiration, and then you're still blowing out, but less flow at a given volume went out. And this is a flow volume loop. It's kind of like a flow rate. And there's a certain slope here by which you went down to your residual volume, telling you how fast the air escaped. So that A was like a maximum, blow it off in a maximum kind of fashion. They tried as hard as they could. In C, person filled their lungs up with air and then they just kind of breathed out in a submaximal way. Well, look, the slope was the same going down to the residual volume. And in B, what they did, they said, hey, you know what? Fill your lungs up with air. I'll just breathe out just a little at first. Right now, I'll speed it up. Then the air goes out. But it's the same slope. Expiratory flow is independent of effort. The airway and the bronchioles pinch off and air gets trapped. It happens in all of us. And it doesn't matter how hard you try to blow off more air. Here's pre-inspiration, inspiration during, and expiration, forced expiration. If you go from pre-inspiration, this is atmospheric air, this is alveolar pressure. There's no airflow because there's no gradient in pressure. The intrapleural pressure is -5. The diaphragm contracts, the intrapleural pressure falls. There's a gradient from atmosphere to the alveolar pressure, air flows in. There's a transmural pressure here. The difference between the intrapleural pressure and the alveolar pressure, and that's allowing the airway to be competent and allow for this delta transpulmonary pressure to enter, let volume enter. At the end of expiration, now we filled up the alveolus with a whole bunch of air, and we're not fully expanding the intrapleural pressure anymore, and over time now, its pressure um, is or stop drawing the breath because it's in equilibrium with the atmosphere. And then we go to do that forced expiratory maneuver like in A, where you push it on out, like your abdominal muscles are pushing it on out. And as the abdominal muscles are pushing on the diaphragm and on the thoracic, on the belly and on the thoracic cavity, now the intrapleural pressure is no longer negative. It's actually a positive pressure, so it's higher than atmosphere. Yes, that's pushing on the alveolus, and you're getting now a reverse transpulmonary pressure. Push, 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 push. But sooner or later, the intrapleural pressure, being now positive, is higher than the transpulmonary pressure. So now the wall gets pinched off and the air will get collapsed. I'm sorry, the air will get trapped in the collapse. This is a figure from the West book. It's fine. I don't love it in terms of the point. I'm actually going to it's the same figure. I'm actually going to go to the next slide, which makes the picture, I think, a little more clearly to the seminal point that at expiration, which is passive, versus forced expiration over on this side, what you're seeing, this is an alveolus and this is the airway. Think of these as the bronchioles. And you're seeing that the alveolar pressure is positive relative to atmosphere. Air is going to flow out. And that is because of the elastic tissue and the surface tension. And we have a nice negative intrapleural pressure. But as we get closer to the mouth to expire the air, sooner or later, we're going to have a lesser driving force through the lung, and we're going to get air trapped because we pinch off the airway, if you will. Now, look what happens in the case of a forced expiration. Now the belly's pushing on the thoracic cavity, making the intrapleural pressures positive. Instead of -8, it's plus 25 centimeters of water. That's making a forward push on the alveolus. And yes, push, 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 push. But now the alveolar pressure is overtaking the transpulmonary pressure, right where these blue lines are landing. And if this pressure is 25, and inside the lung it's 24.9, this pressure is transmurally going to compress on the bronchi, and air is going to get trapped. So that's the main point, is that the more intrapleural pressure, the greater the air trapping. And back to this slide in red, the idea is you need to think about the intrapleural pressure's gradient on the alveolar pressure as the driving pressure. The alveolar pressure minus the intrapleural pressure is really the transmural pressure, the pressure across the wall. That's what's dictating the driving force to push the air out. The harder you try, the higher the alveolar pressure, the less apt you are to not pinch off the airway. Hence, it's an effort-independent phenomenon. So, airway closure is occurring in all of us, and it's worse in disease like emphysema. Now, emphysema has lots of air trapping, and it could become problematic because as those grapes / the analogy to alveoli sort of slough down and get they lose their traction. Their bronchi also start, like the whole grape cluster starts to sag. It's easier to push and pinch off that airway. So they have less recoil, That changes their driving pressure, and it's hard for them to get the air out. So emphysema falls under obstructive disease. Stale air is trapped in their lung, and it's irreversible. Lots of people have emphysema, plus some other obstructive disease like bronchitis, and it's a nasty scenario. So that's I hope you have some appreciation for airway closure and air trapping. The more air trap, the more stale the air, the older the air, if you will, the higher the CO2, the less fresh oxygen. And that's going to impair the way the rest of your body's going to work. Next, I want to cover this idea of resistance in the airway, and we'll talk about some flow patterns. Poussin's law says that the resistance to airflow is, in the numerator, a constant 8 times the length of the airway times the viscosity of the air, like the stickiness of the air. 8's a constant. The length is anatomic, like a big tall person is a long airway, a shorter person is a shorter airway. I mean, it's not really controllable. If we're breathing atmospheric air, the viscosity of the air is the same. You could give different air mixtures with scuba or climb a mountain, those sorts of things. But generally, the numerator isn't anything that's really changing all that much. The denominator, however, is pi times the radius of the airway to the fourth power. Because it's to the fourth power, it's the most powerful determinant of the resistance. So if you've got a narrow tube versus a big old tube, here's the narrow tube, here's the big old tube. Because this is a bigger radius to the fourth power, it has less resistance. This is higher resistance. So bronchodilation would be something that had a lesser resistance. Make no bones about this. The higher the resistance, the harder work there is to do to move air across the lung, whether that be an inspiration or expiration. And the radius of the airway is the most critical factor. And this gives you a little example showing you that a decreased radius by 50%, like in bronchoconstriction, increases the resistance 16-fold. That makes the work of the breathing that much more difficult, that much more vigorous. So that's Poussin's law, very, very important law in all of physiology. Now, one of the things that helps keep the resistance low is air in the lung. This shows you lung volume on the x-axis and the airway resistance on this side of the y-axis in the red line. At high lung volumes, there's less resistance than low lung volumes. Well, think about it. There's more air combating the elastic recoil, combating the surface tension. There's less resistance here. The air is it's maintained the radius as larger, less resistance. That's good. That makes it easier to draw in the next breath of air. So in emphysema, you have this like floppy kind of big volume down here, but it's like dangling. And this floppy nature makes like the lung more prone to get pinched off. There's actually increased resistance in that type of a situation, even though there's more volume getting trapped distally to that. So like as per our lung closure, the resistance is like at that narrowest phase of the tree, which will end up being the bronchi serving given alveoli. So that's the resistance, of which the reciprocal is the conductance. Conductance is the reciprocal of resistance. As there is greater lung volume, there's a greater conductance of air. And so, again, biophysical terms, You calculate the resistance on the Poiseuille's law, and you can express it in resistance, as I will do throughout our discussions together. Most literature will, but some literature will call it conductance, and it's important for you to appreciate what that is. This is showing you the point on the emphysema and the radial traction that I drew, but this is obviously a much nicer figure, showing you a normal lung in the black and an emphysematous lung in that blue hatched line, looking at the lung volume, airway resistance. And let's just take a set volume. The normal line, the normal relationship for the normal lung would have this much resistance at a given lung volume. An emphysema would offer this much more resistance. Why? Back to the analogy of the floppy grapes. Here's an alveolus. Here's the airway wall. It's tethered by like this little elastic tissue setup with like little slinkies, little springs on the side. So the spring like pulls the airway open, like in the direction of these arrows. It's maintained. What happens in emphysema is you lose the integrity of this elastic tissue. It gets floppy, like the rotten grapes. The airway gets collapsed. Greater resistance, lower radius. The greater the resistance, and it gets to be a little bit of double talk, but the greater the air trapping. It's easier to trap air distal in the lung when there's a greater resistance, because you're already that much more along the way of pinching off the airway. So this is a problem. Volume in the airway lowers the resistance or increases the conductance, however you want to spell that out. Now, in the whole airway, here's some factoids on this. Up to 50% of the resistance to airflow is in the upper airway. The other 50% is everything downstream. So from the nose down to the larynx, that's where most of the resistance lies. If you close off the nose or close off the mouth, there's less of an avenue. There's more resistance. If you leaned on the neck or, the oral pharynx in any way or shape or form, like we saw with some of the policing action in the Chauvin trial, yes, that increases the resistance. Biophysically, it increases the resistance. It makes it more difficult to breathe, as per Poiseuille's law. So, when you see folks that are, pathologists and whatnot, not acknowledge that, I just kind of scratch my head, because as a physiologist, it certainly makes it much, much, much more difficult to breathe, both on inspiration and expiration. Now, there's also your vocal cords that open and close, and you've got your stabilizing muscles in the oropharynx that will contract during normal inspiration that can help dilate and stabilize the upper airway. Keeping the upper airway open reduces the resistance, and it makes it easier to breathe. That's the point of number 3. During deep forced inspirations, it's actually very interesting and kind of paradoxical, but the negative pressures that you generate in your thorax can actually cause a sinking mechanism on the upper airway, like the upper airway gets sunk into the chest. Well, that increases the resistance. That's not good. And we've got mechanisms, reflex contractions of pharyngeal dilator muscles that help, keep the airway open and reduce that resistance. But, also of issue. And the smallest airways individually have the smallest resistance because they've got the smallest radius. But because you got so many of them in parallel, the resistance as you go deeper down into the lung gets lower, lower, lower. And it's much higher, like I said, in the upper airway where you have less of these. So resistance is mostly on the top, lesser on the bottom. Yes, there's a smaller radius on the bottom, but there's so many more of them that in parallel, the resistance actually goes down. And we'll talk more about that principle. Not only the anatomical distribution of the ventilatory tree and air will dictate resistance. In the bronchi particularly, which are very much made up of smooth muscle, the smooth muscle along the airway can contract. So you see here's a normal airway, smooth muscles like this pinkish part, and here's a spasm of the smooth muscle where it contracts. Look what happened to the radius. The radius got smaller because the smooth muscle contracted. So bronchoconstriction, as is illustrated right here, would increase the resistance. That's independent of airflow. That's independent of the anatomical regulation or the way it dictates the overall airway resistance. This is like what I call functional airway resistance. And I talk about this on a Lightboard presentation. Plug and chug this little link into your browser and you can, watch a discussion on this. But what is illustrated here by the table is that you've got a lot of factors that cause constriction of the bronchi. Parasympathetic autonomic activation, acetylcholine, prostaglandin, allergic reactants, histamine, alpha adrenergic agonists, alterations in this is a typo, decreased PCO2 in small airways can cause constriction. And then generally the reverse will occur, as is illustrated right here. One of the things I'll just comment on, like asthma, very famously causes bronchoconstriction and air gets trapped in the alveoli. So albuterol and your inhaler is a sympathomimetic beta agonist, beta receptors, beta 2 receptors all along the smooth muscle in the airway. That causes the smooth muscle to relax, make the radius bigger, lessen the resistance, makes it easier to push the air on out. So these are important overall factors to appreciate the functional airway resistance, kind of like the physiologic airway resistance, as opposed to, quote unquote, the anatomical airway resistance and the volume restrictions. Another biophysical factor that I just want to mention is wall tension. This is separate and distinct from resistance. Wall tension is defined by Laplace's law, where the wall tension is the pressure in the airway or in a given alveolus times the radius divided by 2. So if you've got an alveolus, for example, think of a balloon. Here's a normal one, and then the balloon gets like really dilated. Like this balloon over here is more apt to rupture, to burst. That's kind of like what the wall tension's telling you. And so when the radius gets really big and it has a high pressure, that can cause like a rupture, a burst, a barotrauma. We talk about Laplace's law in aneurysms, ventricular aneurysms, in vascular aneurysms. Because you can rewrite Laplace's law for pressure, it also pertains to distribution of air across the alveoli. So what they've done here is they wrote as per pressure, and if you write the tension, rewrite the tension equation to have a pressure on the left side, what it shows you is that because you're able to distribute air across adjacent alveoli, if you had a small alveolus, it would be prone to dump air with a higher pressure into a larger alveolus, making it even smaller, making it collapse. Now, we don't really do that, but on a theoretical basis, Laplace's law helps us think about that as well. One bottom line common sense thing is we, in all of our examples, don't want alveoli to close. Atelectasis is a bad thing because once you get that wind knocked out of you, so to speak, it's so much harder to reinflate that balloon. And Laplace's law is something to greatly think about. And then the last biophysical factor in our discussion today is Reynolds number, which talks about or qualifies, it actually quantifies laminar flow, which is layered flow, versus turbulent flow, like you see in an airplane, those sorts of things. So Reynolds number is calculated by this equation, 2 times the density of the gas times the velocity of the gas, the radius of the tube, divided by the viscosity of the tube. Different gases have different densities. Helium is different than hydrogen, different than nitrogen, different viscosities, the stickiness of gases. Air moves at different velocities, and then there's different tubes. And physicists calculate this stuff. You can calculate Reynolds number, and then based upon that, you can see is air moving in a laminar fashion or a turbulent fashion. From a clinical basis, you can listen to lungs. Strider phenomena would illustrate a high Reynolds number, would illustrate high turbulent flow, which often occurs around transitions in the airway and across narrowed openings across the airway. This shows you layered flow. Air flows fastest in the middle, and it tails off as you get near the end, or the edges, I should say, of the airway. Think of a river. Water flows more slowly at the edges than it does in the middle. Turbulent flow is non-productive flow, like it's not moving that way like you wanted it to get down to the alveolus. It's bumping against the sides. The more turbulent the flow, the more it bangs against the lining of the airway, the epithelial cells, and that can induce barotrauma. This is not productive. And then the truth is in the middle. You get a little turbulence around the sides when you go to transitions, but there's also some layered flow as you get deeper and deeper into it. And if you look at the airway and the anatomy of the airway, you see that there's a whole bunch of turbulence right in this region. These are the Reynolds numbers in the glottis, the trachea, the upper bronchi, at different flow velocities compared to the rest of the system. The table, way back when, 1961, physiologic reviews, but maybe you want to look at it like this. The turbulent flow is highest where the resistance is highest in your anatomy, up around the mouth, the upper layer of the trachea, the primary bronchi, greatest turbulent flow. It's high flow. It's like an expressway, moving a lot of velocity, banging along the sides. And then you get into a divergent, lower resistance in parallel system as we get deeper and deeper and deeper, and the flow pattern becomes more of a laminar flow. So I think we covered quite a number of important topics in our discussion today, and it would be nice now to go back and look at the objectives and sort of repeat some of these issues to yourself. What are the different subsections of pulmonary physiology? Today we talked about number 1, which are the mechanics of breathing. Can you discuss the muscles of breathing? Can you discuss how that affects pleural pressures? Can you discuss how that impacts the work of breathing? And what are the subfactors associated with the work of breathing? How does compliance play into that? How does compliance play into hysteresis? What is hysteresis? What are the factors that induce it? How does surfactant play into hysteresis? How does surfactant play into elasticity of the airway and airway closure? What is surface tension? How does surface tension impact the work of breathing? How does that factor in expiratory maneuvers in airway closure and compression? And how do you think about airway resistance and flow rates and Reynolds number and laminar flow and Laplace's law and those biophysical features into practice, into the use of ventilators, into setting flow rates, etc.? How would a ventilator with a long tube entry into the patient be different than a shorter tube? Why, when you snorkel, do you use just a relatively short snorkel when you could see a lot more fish if you had like a 10-foot snorkel? Why are snorkels set up? I guess they're about 3 feet or so. Why are they set up that way? Does that have something to do with resistance? How do chest tubes play into intrapleural pressure? How to use them clinically? What is that manipulating? So I think thinking about natural breathing, innate breathing against mechanical breathing across the partitions of alveolar pressures, pleural pressures, etc., is where this information can go. And I thank you for thinking of these important issues, and we will stop there.